Diagnostic method in virology is divided into three groups. The first is the direct examination of the specimen, and then indirect examination and serological identification and classical techniques and new techniques. In direct examination, the electron microscopy, morphology, and light microscopy, histology, appearance of inclusion bodies, antigen detection, immunofluorescence, ELISA, and molecular technique for detection of a viral genome. In the direct antigen test, which is under the direct examination, like rapid diagnostic method, RSV and cytomegaloviruses and immunocompromised patient. The nasopharyngeal is spirochetes from respiratory viruses and RSV and flu A and flu B and adenoviruses, detection of rotaviruses antigen in the feces, and cytomegalovirus PP65 antigema test, detection of herpes simplex and varicella zoster in the skin scraping, and hepatitis S antigen in the serum. But usually this technique is tedious, results are difficult to interpret, and sensitivity and specificity is poor. Electron microscopy, in which the virus particles are detected and identified by its morphology, and magnification power of electron microscopy is 50,000 and normally used as 10 raised to power 5 and 10 raised to power 6. Mainly used for the viral gastroenteritis by detecting the virus in the feces, like rotaviruses, adenoviruses, astroviruses, calciviruses, and norovirus like viruses. Occasionally may be used as viruses in the vesicle and other skin lesions, such as the herpes viruses and papilloma viruses. The sensitivity and specificity of EM may be enhanced by the immuno electron microscopy, and in which the virus particle are incubated with specific antibodies in the use of agglutination virus particle, and thus easily capture virus particle into electron microscopy grid. The main problem with the EM is expensive maintenance, and sensitivity is also poor, with at least 10 raised to power 5 or 10 raised to power 6 viruses particle per ml. That's why it is least widely used. In light microscopy, which is also in direct examination, in which the replicating viruses often produce the histological changes in many cells, like inclusion body collection of a replica viruses particle either in the nucleus or cytoplasm, like Negri bodies in the case of rabies, and cytomegalovirus, cytomegalic inclusion bodies in cytomegalic viruses. Viral genome detection, like molecular method dot blot, which is mostly used for the protein, also called the Western blot, and southern blot, in which the DNA, specific DNA and RNA probes for hybridization, and northern blot is most widely used for the RNA. The PCR is most sensitive technique, and ligase chain reaction amplify the probe molecular rather than in nucleotide amplification. Two probes are ligated through ligase enzyme. It's more specific than the PCR. And nucleic acid base amplification, like flu A and FMD, SARS, human barcoviruses, also trypanosoma brucei. All of them have some form of amplification, like bDNA, branch DNA to measure the viral load in HIV sample for quantitative quantification of RNA. It's accurate enough to identify a few as 25 viral particle in early stage of infection. Viral isolation in indirect examinations are through cell lines. When we cultivate the viruses in artificial cell line, most of the cell lines are very tedious technique, and because of the contamination, bacterial contamination or fungal contamination, and also taking so much long to isolate viruses. And we have using the three type of cell lines. The one is primary cell line, which is monkey kidney or normal cell obtained from the freshly killed animal. This primary cell line availability is very hard, and it is very expensive, and wide range of the viruses can grow on these cells, but the passage are once or twice, so we cannot preserve the culture for the long time. Semi-continuous cell lines have the 50-time passage cell line, and human embryonic kidney are skin fibroblast mainly taken from the embryo's tissue, not supported the wide range of growth of the viruses. Continuous cell line, their passage are indefinitely because they are a tumor cell lines, and limited range of the viruses can grow only. These are mostly HeLa, Viro, HIP2, LLC, and BGM and MK2. Identification of growing viruses can be done by the cyclopathic effect, and cell disorder effect may be specific or non-specific, such as in cytomegaloviruses and herpes simplex viruses. The specific cytopathic effect are observed, while enteroviruses do not produce the cytopathic effect. In indirect method, the heme absorption in which the RBC attach it have ability to stick to the mammalian RBC, mainly used for the influenza and para-influenza viruses. Identification of a virus is like neutralization, heme absorption, inhibition, immunofluorescence, and molecular test. The problem with the cell culture are the long period it takes. The 4 week sensitivity is low. Many viruses not grow, like hepatitis B, C, and parvoviruses. Toxicity and the bacterial contamination. Some diarrheal viruses are also unable to grow on the artificial cell line. The rapid culture technique, in which the viral antigen detection is used like 2 to 4 days after the inoculation. The detection of a virus in early stage, some of the viral antigens can be detected like shell wall culture or cytomegalo deep test detection of early antigen fluorescent foci. Inoculation, basically we inoculate and the specimen and rotation 1 to 2 hour, and then viral absorption occur and incubate for, and after the incubation the slide fluorescent technique is used in microscopy. A viral particle can be observed. The mump viruses on infected cell surfaces produce the syncytia, which indistinguishable from that of respiratory syncytial viruses. Further, there is an explanation for the DNA viruses and RNA viruses. In the DNA viruses, herpes simplex viruses, Varicella zoster virus, cytomegaloviruses, Epstein-Barr viruses, adenoviruses, papillomaviruses, and papillomaviruses. And RNA viruses, enterovirus and influenza viruses, parainfluenza viruses, mumps, measles, respiratory syncytial viruses, rabies, and arboviruses, adenoviruses. First, we will explain the DNA viruses. In the DNA viruses, herpes simplex virus is the easiest for cultivation. It will only take 2 to 3 days to appear the cytopathic effects. And the Viro, HEPA, human diploid, rabbit kidney, human amnion cell line can be used. A typical typing by the immunofluorescent neutralization and radio enzyme assay, and direct immunofluorescence for the vesicular cell smear, and electron microscopy for the vesicular cell line, and Varicella zoster are smaller, and human herpes simplex virus is larger. Serological rapid test are complement fixation test, neutralization, immunofluorescent ELISA, and radioimmunoassays. The primary infection can be diagnosed by these antigen detection or antibodies detection test. Varicella zoster virus is only isolated in human diploid cells like HeL and HEC. Typical cytopathic effects are produced by these viruses and difficult to isolate because it is labile, and vesical fluid must be inoculated as soon as possible after the collection, and electron microscopy of vesicle cell smear and direct immunofluorescence for vesicle cell smear. Serological complement fixation test is not sensitive. In case of Varicella zoster viruses, ELISA and immunofluorescence may be used. In cytomegaloviruses, the only human embryonic lungs fibroblast, which is MRC5 cell lines are used 2 to 4 weeks for appear the cytoplasmic characteristic. The rapid culture methods such as the DIF test may be used to yield the positive result within 48 hours. And cytomegaloviruses antigema test, it is roughly detected the several infection by the number of positive cells, neutrophils, PP65, cytomegalovirus antigen. Quantitative PCR for CMV, DNA, and serology complement fixation test again is very low sensitivity. ELISA and lytic agglutination may be used in case of cytomegaloviruses. In Epstein-Barr viruses, the isolation can be done by the blastoid cell lines expressing the EMB antigen. Hybridization and PCR may be used to demonstrate the presence of EBV genome in the biopsy specimen. Indirect immunofluorescent viral antigen detection can be also helpful. In adenoviruses, the isolation can be done on HEPA2 and HEC cell line. Cytopathic effect usually came in 2 to 7 days. Identification of by immunofluorescence and neutralization can also be used. In serology, the complement fixation test is not sensitive, but microneutralization are more sensitive and specific tests available for adenoviruses. For papillomaviruses, we can use the hybridization, PCR biopsy, electron microscopy, immunofluorescence of the lesion scraping, but serology response is low in human papillomaviruses, so it's of no use. In pyelomaviruses, the isolation on BKV isolation or HEK or human diploid fibroblast cell can be used, but the diagnosis is actually made by the cytology or a cytological study of the lesions. In RNA viruses, enteroviruses are isolated. Actually, polioviruses can also be isolated on MK and BGM and LLC and MK2 applied and Viro HEPA2. Coxa viruses, some strain will grow on RD, rhabdomyosarcoma, and some on newborn mice, while Cox B can be isolated on MK, BGM, and LLC, MK2, and Viro HEPA2. Ecoviruses can also be isolated on MK, BGM, and LLM, MK2, human diploid, and RD. Isolation may be identified by neutralization, such as the Lim-Benesch-Melnick A to H pool will identify 42 enteroserotype, but if the single identification cannot be made, then the isolate may be a mixture. In serology, the neutralization is more specific and sensitive method. Metabolic inhibition tests like color change, pH, and pH change or by acid production can also be used. Plug reduction test and but enteroviruses PCR is most widely used, especially in case of CSF during meningitis causes by enteroviruses. Influenza viruses isolation on MK and LLC and MK2 and MDCK. Influenza B viruses produce cytopathic effect on MDCK, monkey kidney cell lines, and viruses detection mainly by the HA antigen identification by IF and HAI test. Serological complement fixation test can be used for the new viruses subtypes. Parainfluenza viruses isolation can also be done by MK, LLC, and MK2. Lewis leukocyte carcinoma, monkey kidney 2 cell lines, and serology. In serology, complement fixation test is non-specific, so we use the heme absorption inhibition test, which is more specific, and also enzyme link immunoassay are available for serological identification of parainfluenza viruses. Mumps isolation may be from the urine, saliva, and CSF on MK and LLC, MK2 and HEC and Viro cell line. They produce the cytopathic effect by syncytial formation. It's a unique property of the mumps, but the cytopathic effect does not occur on the first passage. So heme absorption inhibition should be performed, and confirmation by the uh, inhibition of aminofluorescence and heme absorption inhibition. In serology, the antibody to S antigen will appear in after a few days, while those V antigen uh, with 2 to 4 weeks it's appear. In the measles, the isolation is on MK and HEK, and cytopathic effect takes weeks or more time to appear. Examination is done by the heme absorption inhibition and aminofluorescence. The cytopathic effect consists of multinucleated giant cell, which is the unique property of measles, and definitive identification required the neutralization test. Isolation from the throat swab and conjunctal swab and urine. Direct detection of multinucleated giant cell may be found by the direct examination of nasopharyngeal or urinary sediment smear by the aminofluorescent technique. Serology can be done by CFT and heme absorption inhibition. Widely used in heme absorption inhibition is more specific in case of measles also. Respiratory syncytial viruses are basically isolated on HEPA2, and it is the most sensitive cell lines for respiratory syncytial viruses because it produces the characteristic cytopathic effect on these cell line. But if not produced, you can use aminofluorescent and the direct detection, and ELISA can be also carried out from the nasopharyngeal aspirate. Serological test is also complement fixation test and ELISA, and it is hard to find the antibodies in the acute phase. So in the rabies, the isolation is on intracerebral inoculation on lab mice is the method of choice and most effective method. In direct detection, the Negri bodies are examined. In direct detection, Negri bodies examinations, rabies antigen by aminofluorescence, and the specimen of choice is neck skin biopsy, which is most sensitive than coronal scraping. In serological identification, the mouse neutralization test is the standard method. ELISA is also available, and serum and CSF specimen may be used. Arboviruses is usually present in the blood during the acute febrile phase illness to reduce the effect of the serum inhibition. That's why the dilution of 10 raised to power 5 and 10 raised to power 10 or higher is used. In encephalitis also, in some cases, the brain tissue biopsy is used to culture on cell lines to detect the arbovirus. For arboviruses isolation, three systems are used, like intracerebral inoculation of a new mycetes tissue culture on LLCMK2 or viroarthropod cell line, and intrathoracic inoculation of arthropod. Identification can be done by aminofluorescence and enzyme link immunoassay, and direct examination. We use the aminofluorescence to detect the antigen in the tissue. Serological, we can use heme absorption inhibition commonly used, but also aminofluorescence, radioimmunoassay, and neutralization test also can be used. In arenaviruses, the isolation from the blood during the acute febrile phase of illness, the standard procedure is inoculation on the viro cell line followed by aminofluorescence, and direct detection possible but not used routinely because of need of category 4 contaminant facility. And serological diagnosis is aminofluorescent, complement fixation, neutralization. ELISA may be used. In mycology, Colco4 white stain is excellent fluorescent stain used for detection of fungi in the specimen, and KOH 10% is also used in the gram staining is most useful in mycology for detection of aerobic actinomycete, but for other yeast and fungi, morphological features are not clear in the gram stain. Modified canyon or acid-fast stain is used for the partially acid-fast filament of Nocardia species. In right stain and Giemsa stain is used for the intracellular yeast like Histoplasma capsulatum in the blood and the bone marrow, and periodic acid Schiff pass and glomerulomethionine silver stain is used for demonstrating fungi in the tissue. Rice grain sloop medium is most widely used to identify the Microsporum species like Microsporum odani produce the brown pigment and sporulate and produce the rocket slope hyphae in hairs, while the M. canis do not produce the pigment and sporulating in the hairs. So for M. canis, SDA in mycosal is used for SDA and mycosal. M. canis produce the bright yellow pigment. M. canis lesions with the hair loss and hair show the fluorescence under the dark microscopy. The hair proliferation test from the pigs that penetrate into the hair shaft, commonly used to differentiate between the dermatophyte. Bread seed agar, in which the nigger seed and penicillin 20 microgram unit and gentamicin 40 mg per ml is basically used for the differentiation of Cryptococcus neoformans from the Cryptococcus gatti. Cryptococcus neoformans produce the brown black pigment with the smooth colonies on this media. Bromocerasol purple milk solid glucose agar is used specialized medium for the Triptophyton species. Triptophyton species are known for the dermatophytosis, and they are also known as the ringworm, pinworm, and hookworm. Redness, itching, scaly, circular rashes, hair loss may occur in the affected area. This dermatophytes, trichophyton species are also called the tinea species because they produce some infection like in athlete foot are also called tinea pedis and tinea corrosus are grown ringworm, and tinea onginum is a nail infection, and tinea munum is hand, and tinea corpus is body, and tinea capitis is scalp, tinea facie face, tinea baby is beard, and tinea niger is black. Creatinine, dextrose, bromothiamol, butyramine is basically used for differentiation between the Cryptococcus species. And serotype D, which is Cryptococcus neoformans, produce the bright red colony, turning the medium into bright orange after the 5 days inoculation at 28 degrees centigrade, while serotype A, which is Cryptococcus neoformans Gurbi, produce no change, whereas the group B and C causes the slight greening of the medium. Cornmeal agar is used for routine culture identification of a fungi. Cornmeal glucose sucrose yeast extract agar is used for stimulating sporulation in some zygomycetes like Saxenia and Apolyzomyces. And Chiffic dox agar is used for the water and wastewater for isolation of Aspergillus, Spencerine, and Candida. And modified Dexon agar for the for twin peck is basically used for the normal flora of the skin, that is Melissa furfur. It's a yeast and mold, and cultivation and maintenance can be used and naturally found in the skin surfaces. Here, proliferating test for dermatophytes, trichophyton mentagrophytes is differentiated on this test because the organism produce the mark erosion, and while the T. rubrum does not cause any erosion in the hairs of pubital hairs. Lectrimyl agar is used for the trichophyton rubrum, which produce the pigment on this medium, and Letman oxgall agar is used for the inoculation of skin, nails, and hair. The malt extract agar and 1% peptone water Peptone agar is cultivation and differentiation of fungi. PDA is a general purpose media in mycology. SDA with antibiotic and chloramphenicol, gentamicin and yeast extracts and cyclohexamide is used for the dermatophytes. SDA with the chloramphenicol, gentamicin, yeast are used for yeast and mold without the cyclohexamide, because cyclohexamide is inhibition is also inhibited some of the molds and yeast form of the fungi. SDA with the 5% salt differentiate between the T rubrum because and T mentagrophyte, because the T rubrum produce the red pigment while T mentagrophytes are produce no pigment. The tape water agar is most widely used for stimulating the sporulation in some zygomycetes like of apoplasomyces and Saxena isolates. And urea agar sloop with the 5% 0.5% glucose is used to differentiate between the different urease production organisms like trichophyton, aspergillus niger and coccidioids are urea positive. Vitamin free agar is used for trichophyton agar, trichophytons, basically it is also called vitamin free agar and trichophyton agar number 1 differentiate between the trichophyton species. If the medium plate if the coccidiod is suspected, the culture plating method is not used because it's very dangerous, because the organism is very contagious and can be inhaled through aerosol. The identification of yeast are first done by the germ test tube method, in which the mycosal production, also known as feeds in case of Candida albicans, while the Candida dubilensis are negative. Albicans tropicus and Kusri is identified on 48-hour chrome agar, while the while in C. galbata, 1 hour test known as rat test, in which the telerose assimilation is checked, and this is the unique property of only Candida galbata, which produce a telerose assimilation. Mucoid colonies on PDA, we suspect the C. neoformans or C. gati. Whitprip in India Inc. confirmed the capsule and urease slant inoculation and cron meal 20 tween 80 agar glucose free is also used to differentiate between the C. neoformans and gati. And Further, phenol oxidase or caffeinic acid disk test can also be used for the primary colony, because of the dextrose present in the medium. Sometimes it gives the negative results. If the colonies are not mucoid, the germ test tube is used. If it is negative, if it is positive, then C. albicans is suspected, which is positive in the 3 hours produce the feeds. And if it is negative, then we go for the slow growing bacterium. Like first we rule out the C. galbata by rate test. If it is negative, then use the commercial kit for the yeast identification or chrome agar. Sporulation inducement method for the apolysomices and Saxena species, which is zygomycetes. Apolysis are filamentous fungi cause the Apophyllis elegans mucormycosis, and Saxena are also zygomycete, but it's are flux-shaped sporonium with the spherical venter. We can stimulate the sporulation up to 5 to 7 days at 42 degrees centigrade for all non-sporulating zygomycete from the clinical sample primary growth, and SDA Kate 1 centimeter agar with the fungal growth, and place into the sterile distilled water with the yeast extract. Incubate 37 degrees centigrade for 5 days and make wet form from distilled water and observe the sporangia. As we explained previously, in the dermatophyte family, the Trichophyton mentagrophyte and Trichophyton rubrum is differentiated on the basis of hair proliferation test. where the mentagrophytes are positive. Actually, it is taken from the skin and scalp, while the T rubrum is negative. It's usually causes the elite foot disease, ringworm, and jock itch. 1 centimeter long fragment of a healthy human hair is used in the petri dish and inoculate both organisms and check for the positivity after 12-hour incubation. Black yeast. Actually, Drysolaria and Biopolaris and Exocerohilum groups are also called the black yeast because it's very pathogenic organism and very slow growing organism, especially pathogen of the plant, especially for the grasses. They have the dry spore. They cause the type 1 hypersensitivity, asthma, and high fever, and also cause the pilohyphomycosis, include the keratitis, sinusitis, and osteomyelitis. They produce the gray to dark brown colonies, and their spores are very dry. Identification can be made by tape method, and they are also called the black yeast or diatomaceous fungi or microcolony fungi or meristematic fungi. Slow growing microfungi, mostly asexually, mostly resistant organism up to date. That's why called the rocky inhibiting fungi. The rock inhibiting fungi like E. dermatitis and Hortina wernicki is a mold organism used for the bioremediation for a polluted ecosystem. The germ test tube is widely used to differentiate these three black yeast from each other. In the germ tubes, the Biopolaris, as the name indicate, have biopole, and Drosera have only single pole, that is on left side, and exohilum is long and elongated. For identification of Nocardia species, we use the acetamide agar. In acetamide agar, the red color indicates the presence of Nocardia, while the yellow-orange are considered as negative. Aryl sulfatase broth or Middlebrook 7H9 broth supplemented with the substrate potassium phenolphthalein also produce the same color change. If it is red, then it is considered Nocardia species, or if it is no color change, that is considered negative. Ascospore media is mostly used in the slants, and medium for the ascospores are V8 vegetable juices medium. Assimilation media for the yeast assimilation is the utilization of a carbon nitrogen source by the microorganism in the presence of oxygen. A positive reaction is indication of the presence of a growth or a pH shift in the medium. Wickham broth method is a carbon assimilation media, yeast nitrogen base carbon and distilled water. Nitrogen assimilation media is yeast carbon base, potassium nitrate, and distilled water. The brain heart infusion with the sheep blood is added, and this is widely used to isolate the histoplasma capsulatum in blastomyces dermatitis. The buffer charcoal yeast agar is basically used for the cultivation of Legionella species, but also used as a recovery of Nocardia species, and it is further supplemented with the vancomycin and